students, in the previous topic we have studied about absorption of water by root. Root absorbs water and minerals from the soil through the xylem vessel. It rises up to the leaves where partly water is used in the photosynthesis and another life processes. The excess of water is required to be eliminated, and the maximum amount of water is lost due to evaporation from the free surface of the leaf as well as from the other parts also. The loss of water in the form of water vapor is known as transpiration. So it plays important role in the life process of the plant. So what is transpiration? The loss of water in the form of water vapor from aerial parts of the plant is called transpiration. In short form, it can also be written at some places. You will find this type of definition also, that is the evaporative loss of water. Evaporative loss of water from aerial parts of the plant is called transpiration. Means the water has to be lost in the form of water vapor. Now the question arises, is there any other method also by which the water is lost from the plant? Yes, this is called guttation, which we had studied in previous chapter, as well as you will also define in this process, where the water is lost in the form of droplet along the margin of the leaf. So here it is transpiration. Now transpiration occurs in different forms, that is from different parts of the plant. We have to study first of all the types of transpiration, then we will study about the experiments demonstrating the process of transpiration and to measure the rate of transpiration. So here we have to study here, that is types of transpiration. But before going to study the types of transpiration, we should first of all study the difference between dicot and monocot leaves. There are two types of higher plants. Dicot, that is in the seed there is two cotyledons, and monocot, that is the seed has single cotyledon. So here first of all we will study this difference, because we have to answer some related experimental questions on this basis. So first of all study the difference between isobilateral leaf, which is also known as monocot leaf, and second dorsiventral leaf, also known as dicot leaf. Now first of all you see isobilateral. Iso means same, means both the surfaces of the leaf will be the similar. Here dorsiventral means the two surfaces, that is the upper and lower surface, have different structures, different compositions. So there is dorsal as well as ventral surface. So first of all start along with its different topics or points. First point, about the arrangement of leaves in the plant. In dorsiventral leaf, the leaf is arranged vertically such that both surfaces are equally exposed to sunlight, equally exposed to sunlight. That is, in this case the leaf will be arranged like this. This type of arrangement will be there. This is the vertical arrangement such that it will be exposed to sunlight on both surfaces equally. But here the leaf is arranged horizontally at right angle with the twig such that upper surface is exposed to sunlight. Exposed to sunlight means its upper surface will only receive sunlight. Like you see, suppose here is the branch, twig, and this will be the arrangement of the leaf. Now such that this upper surface is only exposed to sunlight. The lower surface will be, that is on under surface, will not be exposed to sunlight. So there will be some structural difference also, like you see. The another point about its structure. Second point. The both surfaces, in this case isobilateral, both surfaces are equally green. But in this case upper surface is more green and shiny. Upper surface is more green and shiny. The reason is that because chloroplasts are more concentrated in the cells of upper surface. So its upper surface will be more green and shiny than the lower surface. You can observe this difference in leaves of many garden plants. Even you can observe this difference in case of mango leaf and so on. Third, stomata. Stomata are the openings through which most of the transpiration takes place. They are present on the surface of leaves, epidermis layer of the leaves, and young green stems. In this case, stomata are equally distributed on both surfaces because both surfaces are equally exposed to sunlight. So on both surfaces equal rate of photosynthesis takes place. As a result, for diffusion of CO2 there is equal distribution of stomata. But in this case the upper surface is more exposed to sunlight, so chloroplasts are mostly concentrated on the upper surface. As a result, here stomata are more on the lower surface. That is, in this case there is less or negligible number of stomata on the upper surface, whereas there are maximum number of stomata on the lower surface. What is the reason behind it? Because if stomata will be present on the upper surface, since it is directly exposed to sunlight, as a result maximum transpiration will take place and the leaf may wilt. As well as if the stomata will be present on the upper surface, then due to the raindrops, stomata will be closed Stomatal opening will be closed and hence diffusion of gases will be stopped. So there are more stomata on the under surface, lower surface of the leaf. Now the next point. Here, now student we see here, that is in case of monocot plant or dicot plant, there are the upper and lower epidermis layer, and in between the epidermis layer we see that is the stomata, that is in between the epidermis layer the cells are called mesophyll cells. In case of monocot plant, the mesophyll is not differentiated because both surfaces are the same. So here mesophyll, that is the cells in between the upper epidermis and lower epidermis of the leaf. Mesophyll cells between upper and lower epidermis. Lower epidermis. Mesophyll is not differentiated. Is not differentiated. Differentiated means there is no differentiation between cells of the mesophyll. It is equally arranged. But in this case, the upper surface, the cells of the upper surface contains maximum chloroplast. On the lower surface there are more stomata. So here mesophyll is differentiated into upper palisade cells. Palisade cells which are tall cells at right angle to the upper epidermis. Upper epidermis. So mesophyll is differentiated into upper palisade cells and lower spongy mesophyll cells. Spongy mesophyll cells. So this is the difference between the arrangement of cells in the leaf of two types. Now the fifth type. The stomatal opening is regulated by guard cells. Like you see, suppose if there is the leaf and here is the stomata, then this is the guard cell. So what will be the shape of the guard cell in these two cases? Like you see, in isobilateral cell leaf, guard cell is dumbbell shaped. Dumbbell shaped. What does it mean? Dumbbell shaped, like this. Now here there is the stomatal opening. This is stoma. This is guard cell. Guard cell. And this is its inner thicker wall. Now in this case we see that is guard cell is bean shaped, or we write here, that is actually we write here kidney shaped. Kidney shaped. Like you see, this is the arrangement of guard cells. Like this is the bean shaped or kidney shaped, in which there is nucleus and chloroplasts. Nucleus and chloroplasts. Now this is stoma, guard cell, and then around it there will be the epidermal cell. So this is the difference between the two types of guard cells. Now last one. Actually all these differences can be studied in section only. This is the internal structure. But how will you identify morphologically, that is observing from outside, that what type of leaf is it? Then this is the most important point for identification. And what is it? Now here we write about venation. Venation means arrangement of veins, that is vascular bundle, that is the bundle consisting of xylem and phloem. In leaf, from the root, when water is absorbed, through the stem, xylem vessel carries water, it enters into leaves and then get distributed through the tubular, thin narrow tubes called veins. Now, the arrangement of veins is different in the two types. Here, it is called venation. Venation means the arrangement of veins. Venation is, or we can write here like this, there is parallel venation. Parallel venation, arrangement of veins. And what is it? Like suppose here is the leaf, then in this type of leaf there is no midrib, but here this type of arrangement takes place. This is called parallel venation. This is called parallel venation. But here there is reticulate venation. Reticulate venation. So by observing this one, you will be able to identify the type of leaf. Like, unlike this one, here the petiole continues in the middle. That is called midrib. And then from here there are branches, where there is further branches forming network like a structure. So this type of arrangement is called reticulate venation. So this is the point by which you can observe and identify the type of leaf to be studied. Clear, student? Now the next part is type of transpiration. The structure through which transpiration takes place determines the types of transpiration. Like, the first type is cuticular transpiration. Cuticular transpiration. Now, what is cuticle? So, cuticle is the waxy layer on the surface of leaf. Surface of leaf means, that is, on the surface of epidermis of the leaf there is a thin layer of cuticle, waxy layer, through which transpiration takes place. Actually, in desert plants there is thick layer of cuticle to reduce the rate of transpiration, to prevent excessive loss of transpiration. But if the cuticle layer is thin, then some amount of water also evaporates passing through that layer. This is called cuticular transpiration. Clear, student? Now, second is the lenticular transpiration. Now, here again the term comes, that is called lenticel. Now, what are lenticels? Then lenticels are the openings present on the bark of old stems. Now, what are actually lenticels? So if we study the diagram of the stem, now here is the epidermis layer. Now over the epidermis layer there is a thick layer of dead cells. As the upper layer of cells dies, further some excretory wastes are also deposited. So in between this dead layer of cells, some cracks are developed, and these cracks are called lenticels. Water from the living cells exposed to these cracks evaporates that is called lenticular transpiration. So lenticels also help in transpiration. Now the third part is the stomatal transpiration, and through cuticle, through lenticels, a small amount of water evaporates, but the most of the water is transpired through stomata. That is, this is called stomatal transpiration. So, stomata, its singular is stoma. Stomata are the openings present on the surface of leaf and young green stems. Now you see, stomata are mainly responsible for the diffusion of carbon dioxide for photosynthesis. Stomata is for diffusion of CO2, carbon dioxide, for photosynthesis through which water is also lost by transpiration. So most or the maximum amount of water is lost by transpiration through stomata, and actually by this process, that is through stomatal transpiration, it creates suction force, which also helps in ascent of sap, distribution of materials and other things. But before going to describe the mechanism of stomatal transpiration, we should study here the difference between the lenticels and stomata. So we should differentiate here between lenticel and stomata. We will first of all explain its location, distribution. So lenticels, the openings present on the surface of leaf. Openings present on the bark of old stems. So lenticels are openings, just we have discussed earlier, openings present on the bark of old stems, whereas stomata are the openings present along the margin of the on the surface of a leaf. So stomata are the openings on the surface of leaf and young green stems. So this is the first difference related to its location. Now the second point related to its opening. Lenticels, they remain open all the time. Means its opening and closing is not regulated by any structure, but here they open during the daytime. They open during the daytime for photosynthesis. Just we have studied that stomata is mainly to allow diffusion of CO2 for photosynthesis, and through that opened stomata transpiration also takes place. Now the third point about its regulation. Now we know that uh, its opening is not regulated. Its opening is not regulated by any structure. But here its opening is regulated by guard cells. Means its opening and closing can take place. So its opening is regulated by guard cells. As well as further you can see, for the function, through the opened lenticels, diffusion of respiratory gases also takes place. So they help in transpiration up to little extent, and diffusion of respiratory gases. Here you see, that is, now you see here, that is, here photosynthesis also takes place. So they help in photosynthesis by diffusion of CO2, transpiration, and diffusion of respiratory gases. Like you see, during the night when photosynthesis does not take place, at that time the oxygen diffuses in through the partially opened stomata and CO2 is diffused out. So these were the differences between lenticels and stomata. Now here we can study about the mechanism of stomatal transpiration. Mechanism of stomatal transpiration. Now actually this mechanism has to be studied mainly in dicot leaf. So we should draw the diagram of this dicot leaf, then it will be able to describe it more clearly. Suppose this is the upper epidermis layer of cells, and here there is the lower epidermis, but in between there are the stoma, singular is stoma. So when we will have to label, we will mark stoma, but when we will have to describe it in word, we will write stomata. So here it is stoma. This is lower epidermis. Now this is upper epidermis. Diagram based question is also asked. Upper epidermis. Now on the surface upper epidermis layer there are cuticle layer. Now we see here, that is, below this upper epidermis there are tall pillar like cells at right angle to the upper epidermis containing large number of chloroplasts, and actually these cells are called palisade. Below this layer there are loosely packed parenchymatous cells, and this layer of cell is known as mesophyll, spongy mesophyll. Together it will be mesophyll. So we have studied that here the mesophyll is differentiated into palisade and spongy mesophyll cells. Then here there is the structure marked like this, having thick walled cells like this, and then small cells like this, that is xylem and phloem, and together known as vein. So in the diagram this time one single vein is represented here. Now you have to describe the mechanism of stomatal transpiration. What happens here? Now student, we see that is when root absorbs water through the xylem vessel, it rises up to the through the stem and reach to the leaves. In the midrib and then through the veins, water reach to every part of the leaf. From the veins, water reach to every cell, mostly through the imbibition along the cell wall of the cell and partly through diffusion process. So these surfaces of the leaf is moist. So we write here, we explain here how we will start to write mechanism. From the moist surface of the spongy mesophyll cells, water evaporates and saturates the air in the space. This saturated air gradually diffuses into the substomatal space. Substomatal space, that is the space above the stoma. And from this substomatal space, water evaporates into the air. So this is the mechanism of stomatal transpiration. Again remember, when we will have to write, start from here. That is, from the moist surface of the spongy mesophyll cells, water evaporates, saturating the air in the intercellular spaces. This air then diffuses into the substomatal space, and finally from there it diffuses into the atmospheric air in the form of water vapor. So this is the stomatal transpiration. But you see, a student, here that when stomata will be opened, then the transpiration will take place. So we describe here two types of questions related to it. And what is this question? First question here, give reason or explain. First point, transpiration is the price paid for photosynthesis. Transpiration is the price paid for photosynthesis. And second, transpiration accompanies photosynthesis. Transpiration accompanies photosynthesis. So for both the questions, the answer will be the similar. What is this type of answer? Here for this one, again you see, during daytime, stomata open to allow diffusion of carbon dioxide for photosynthesis. Through that opened stomata, water is lost by the process of transpiration. Hence, transpiration is the price paid for photosynthesis. Or we can further write for this one, transpiration accompanies photosynthesis again. That is, during daytime, stomata open to allow diffusion of CO2 for photosynthesis, and through that opened stomata, water is lost by transpiration. So when photosynthesis will take place, then through the opened stomata transpiration will also occur or take place. Clear, student? So this is about the mechanism of stomatal transpiration and the reasoning related to it. Clear, student? Now in next part we will study the experiments related to transpiration as well as demonstration of the process of transpiration by potometer. Clear, student? Okay.